This is Heliox, where evidence meets empathy. Independent, moderated, timely, deep, gentle, clinical, global, and community conversations about things that matter. Breathe easy, we go deep and lightly surface the big ideas. I want you to picture something with me, and you might want to grab a sweater for this because it is absolutely freezing.
Yeah, definitely freezing.
Right. You're standing at an altitude of 4,000 meters. And for anyone listening who lives near sea level, I mean, that is over 13,000 feet up in the air.
Just to put that altitude into perspective for our listeners, that is significantly higher than most commercial ski resorts.
Wow.
Yeah, if you were dropped there right now, every single breath you take would require conscious effort. The atmospheric pressure drops so low that the oxygen molecules are just spread incredibly thin.
So you'd feel it immediately.
Oh, absolutely. It is the kind of thin air that makes your lungs burn, your head spin, and your heart race just from taking a few steps. That is, assuming you haven't spent weeks acclimating to it.
Right, which most of us haven't. So you are standing in the Himalayas. Specifically, this is a region called Ladakh, located in the far north of India. It's this landscape defined by jagged, snow-capped peaks, deep, arid valleys, and just howling, bone-chilling winds.
Intense?
It really is. You are standing on a steep, rocky pasture land above this tiny, remote village, and right in front of you, cut right into the mountain face, is a dark opening.
And it looks less like a cave and more like a jagged scar just ripped into the rock.
Yeah, exactly. The locals call it Abhishranjamo Bao, which, translated from the local dialect, means Old Lady Spider Cave.
Which is a name that serves as a fairly clear warning that this is not a destination for a casual afternoon hike.
No kidding. So you step inside, leaving that howling wind behind, and instantly the darkness just swallows you. You click on your flashlight, the beam cuts through the thick settling dust, revealing a really large entry room.
Right.
The stone walls are etched with ancient petroglyphs, these mysterious carvings of animals and geometric symbols left by people thousands of years ago. You also see the scattered broken stones of what look like looted graves.
Which tells us it's an environment that has clearly seen human activity for a very long time. Though those scattered stones suggest not all of that activity was particularly respectful of the site.
Yeah, clearly. But you aren't stopping in the entry room. You keep moving deeper. You find this narrow tunnel at the back of the room. You have to physically squeeze through it, feeling the rough, freezing rock scraping against your jacket.
And it gets tight.
Super tight. The tunnel opens up into a second large room. Your flashlight sweeps across the floor and you just freeze.
Because the floor of this second chamber is completely covered in human bones.
Densely scattered clusters of them, and then it gets even more claustrophobic. Because from this second room, there's another passageway leading to a third, even deeper room.
Right.
To get in there, you have to crawl. The ceiling is only 50 centimeters high, which is like barely a foot and a half. You are flat on your belly, in the pitch black, surrounded by ancient human remains in Old Lady Spider Cave.
It really feels like the ultimate archaeological locked room mystery.
Okay, let's unpack this. Because what researchers found in that cave isn't just a creepy story to tell around a campfire. We are going on a journey today to piece together the history of a literal ghost population.
It's quite the journey.
It is. Our source material for this deep dive is a fascinating, genuinely groundbreaking scientific paper.
And the genomic data contained inside this paper, it essentially functions as a genetic time machine. It details breakthrough information extracted directly from those bones and incredibly naturally mummified tissues found in that spider cave.
Yeah, the mummies are wild. Today, you and I are going to explore exactly who these people were. We're going to look at a perfectly balanced 50-50 genetic split that completely baffled the researchers. We're going to reconstruct their timeline, and we're even going to track down a ghostly echo of DNA from an entirely extinct species of human.
It's a wild ride.
It really is. But before we get into the mummies and the whole DNA sequencing thing, I think we need to ground ourselves in why finding any of this in South Asia is such a monumental deal for science.
Yeah, that's crucial. It really cannot be overstated how rare this kind of preservation is. Reconstructing ancient population history in South Asia using DNA has, well, it's traditionally been a total nightmare for geneticists.
Nightmare how?
Well, the entire field basically hits a brick wall when dealing with this part of the world. And it all comes down to the underlying chemistry of how DNA degrades in specific climates.
Because DNA is a pretty fragile molecule, isn't it?
Oh, it is incredibly unstable outside of a living cell.
Yeah.
DNA is a complex molecule, and it's held together by a sugar phosphate backbone. When a living organism dies, the repair mechanisms just stop, and that backbone immediately starts to break down.
Okay.
And the two absolute worst enemies of DNA preservation are heat and humidity. If you bury a body in a hot, humid environment, which perfectly describes a massive portion of the South Asian subcontinent, the DNA in those bones will often degrade completely within just a few centuries.
I would assume the heat just cooks it, right?
In a chemical sense, yeah, exactly.
Yeah.
Heat accelerates a process called hydrolysis. That's where water molecules literally cleave the chemical bonds of the DNA strands.
Oh, wow.
Right. It essentially shatters the long genetic code into tiny, unreadable fragments. On top of that, the moisture encourages the rapid growth of soil bacteria and fungi, and they view the organic material inside the bone as a buffet. They literally eat the genetic material.
So, trying to find viable ancient DNA in India is usually like trying to find a snowflake in an oven.
That is a highly accurate way to visualize the problem, yeah?
Yeah.
If you look at the major ancient DNA breakthroughs over the last 20 years, they almost all come from places like the permafrost in Siberia or deep, cool caves in Europe.
Because it's freezing there.
Exactly. In those places, the constant cold and dry conditions act like a natural freezer. It slows down molecular motion and preserves the genetic material for tens of thousands of years. But South Asia has historically been this massive blind spot for geneticists, simply because the environment actively destroys the evidence.
Which brings us back to the altitude of Old Lady Spider Cave. Being 4,000 meters above sea level completely changes the game.
It does. The climate inside that specific cave is freezing and incredibly arid. It bypasses the usual heat and humidity of the lower subcontinent entirely. It basically functions as a natural time capsule.
So let's talk about the excavation itself. The year is 2021. A specialized team operating under the Archaeological Survey of India, or the ASI, decides they need to properly investigate this cave.
Okay.
Now, as we established in the opening, the first room had already been looted. People have known about this cave for centuries. It's not like the ASI were the first humans to stumble upon it in a millennia.
No, not at all. Humans have been visiting the cave, moving things around, and likely taking souvenirs for a very long time.
Right.
And that is precisely why the bones in the second and third rooms were so disturbed and scattered. One of the primary goals of the 2021 ASI excavation was to establish the true archaeological context of these bones. They needed to figure out if there were undisturbed layers beneath the current messy floor of the cave.
And what they found wasn't just old, dry bones. Because of that high-altitude deep freeze we talked about, they found naturally mummified tissues. We're talking skin, ligaments, and muscles still attached to the bone.
But the sheer level of preservation was staggering. I mean, we are talking about human remains from roughly 1,500 years ago, and the biological material is still intact enough to visibly identify the cellular structure of the tissue.
There is this one detail from the source material that I just cannot get out of my head. They found 2 naturally mummified human hands, just like hands, preserved perfectly for a millennium and a half.
Yeah, it's incredible.
And the researchers, knowing they have this incredibly precious, totally uncontaminated ancient DNA gold mine, have to figure out how to transport it down a mountain without ruining it.
Because contamination is the absolute nightmare scenario for anyone working in ancient paleogenomics.
Oh.
Modern human DNA is everywhere. It is on your breath. It is constantly shedding from your skin cells. It is on the fibers of your clothes.
It's practically floating in the air.
Exactly. If a modern researcher even breathes heavily on an ancient bone, their modern, highly intact DNA will completely overwhelm the tiny fragmented amounts of ancient DNA hiding inside the sample.
And that ruins the test.
Completely. When you put that sample into the sequencing machines later, it will just pick up the modern researcher's DNA, rendering the whole expensive experiment useless.
I read how they solved this, and it's amazing. To protect these 1,500-year-old mummified hands from the modern world, they wrap them in kitchen-grade aluminum foil.
I know, it sounds almost comical when you think about high-level science, but aluminum foil is actually a highly effective sterile barrier.
I just love the image of these highly trained archaeologists freezing 4,000 meters up a mountain inside a claustrophobic spider cave, carefully wrapping ancient mummified hands in the exact same foil I used to wrap up my leftover pizza.
It's a funny image, yeah. But there is a beautiful scientific logic to it, though. Aluminum foil is manufactured at incredibly high temperatures. That manufacturing process incinerates any modern biological material on the foil itself.
Oh, that makes sense.
Yeah, so when you pull it off the roll, the inside surface is essentially pristine and sterile. They wrapped the bones in mummified tissues in that clean foil, placed them in clean plastic bags at their research institute in Pune, and then literally hand-carried them onto a commercial flight to a specialized ancient DNA laboratory at Harvard University in Boston.
They were not going to let those bags out of their sight for a second. But I want to pause and challenge the context of this cave for a second. We have men, women, and children all found together in this tiny, incredibly hard-to-reach cave.
Right, a diverse group.
Yeah, we have infants and elders in the same space. My first thought when I read that was, were they hiding? Was there a war in the valley below or a plague, and this family just fled into the deepest, darkest hole they could find and ended up dying there together? Like, was this some kind of tragic last stand?
It is a very cinematic assumption, and a totally natural one to make when you see a diverse group of ages and sexes clustered in an isolated, dark place like that. But the physical archaeological evidence points to a completely different scenario.
Really?
Yes. The key to unlocking that scenario lies in the specific types of bones that survived. The researchers noted highly preserved, delicate skeletal elements, specifically the tiny bones of the hands and feet.
I would assume a foot bone is just a foot bone.
Mm-hmm.
Why do the tiny ones matter so much to an archaeologist trying to figure out how someone died?
It comes down to the biology of how a human body decomposes. When a body starts to break down, the connective tissues, so the ligaments, the tendons, the cartilage, those are the first things to rot away.
Right.
Because of this, the smallest bones, like the phalanges in your fingers and toes, are the very first pieces of the skeleton to detach and scatter.
Ah, I see where this is going.
Yeah, if these bodies had died or been buried somewhere else, allowed to decompose for a few months or years, and then later their bones were gathered up and moved into the cave, which is what anthropologists call secondary burial, those tiny hand and foot bones would almost certainly have been lost in the move.
Because they would have just fallen off.
Exactly. You would just find the big, robust bones, the femurs, the skulls, the pelvises.
So the fact that they found all these delicate hand and foot bones perfectly articulated in the cave means the bodies were laid on the cave floor while they were still fully intact, fleshed bodies.
Precisely. They decomposed, or rather mummified, in the cold, exactly where they were placed by the living. This is a primary deposition.
So it wasn't a desperate hideout from an invading army. It was a tomb. It was a communal resting place for a specific group of people who intentionally carried their dead up into this freezing, dark environment.
They were deliberately brought there to rest. And to figure out exactly when this resting period occurred, the researchers utilized radiocarbon dating.
Carbon-14 dating. We hear about this all the time, but how did they actually do it on these specific bones to ensure they got an accurate date, considering the cave was a mess of dust and looting?
Well, they used a highly precise method called accelerator mass spectrometry on 8 of the bone samples. But before they could put the bone in the machine, they had to go through a rigorous chemical cleaning process. They washed the bone fragments in methanol, acetone, and dichloromethane.
Wait, how does scrubbing a bone with harsh industrial chemicals help find its age? Doesn't that risk destroying the sample entirely?
You're trying to strip away centuries of environmental contamination. If a modern looter touched the bone, the oils from their skin contain modern carbon. If dirt from the cave floor seeped in, that contains different carbon.
Oh, and that would throw the date off.
Hugely. So the chemicals dissolve those modern surface contaminants without destroying the internal matrix of the bone. After that wash, they extract and purify the bone collagen. They want to isolate just the pure ancient protein that formed when the person was alive.
And when they ran the purified collagen through the mass spectrometer, what was the timeline?
The dates were remarkably consistent. The 95% confidence intervals for all the samples overlapped beautifully. They placed this group of people living between roughly 1513 and 1422, calibrated years before the present.
Wow, that tight.
Yeah. And if we look at the absolute widest margin of error in the data, it's between 1690 and 1347 years before present.
So, to round it out for you listening, we are looking at roughly 1,500 years ago. That puts them squarely in the post-Kushan period of the region, somewhere around the 500 CE.
Which means we now have the Where Old Lady Spider Cave at 4,000 meters. We have the a communal primary burial of mummified remains, and we have the Wen 1,500 years ago.
So the next massive hurdle, and the entire reason they went through the ordeal of wrapping those hands in foil and flying them across the world to Harvard, is uncovering the who.
Exactly.
Yeah.
It is time to dive into the DNA. And let me tell you, this was not a simple swab and scan operation.
No kidding. The dataset assembly was a massive technological undertaking. They extracted DNA from 11 specimens in total, right? 8 were dense bones, like a collarbone, a femur, a rib, and 3 were pieces of that mummified soft tissue from the pelvic area.
Correct.
I noticed in the methodology section that they had to use completely different chemical baths depending on whether they were dissolving the bone or the mummified soft tissue. Why wouldn't a standard DNA extraction kit work for both?
It just highlights how delicate and customized ancient DNA extraction has to be. Bone is essentially a hard mineral matrix of calcium phosphate protecting the cells inside, while mummified tissue is a complex tangle of dried proteins and fats.
So they need different keys to open those different locks.
Exactly. You need a highly aggressive buffer to demineralize bone. But that same aggressive chemical bath would completely obliterate the fragile DNA trapped in the softer mummified tissue. They had to tailor the chemical enzymes specifically to carefully break open the different materials without destroying the genetic payload inside.
That makes perfect sense. So they incubate the samples overnight, use microscopic silica beads to magnetically pull the DNA out of the chemical soup, and then they use something called the twist ancient DNA assay. The paper mentions this was crucial. What exactly does this assay do?
To understand the twist assay, you really have to understand the main problem with ancient genomes. If you just take a 1,500-year-old bone, grind it up, extract the DNA, and sequence everything you find, you'll be deeply disappointed.
Because it's a mess.
Because about 99% of the genetic code the machine spits out will belong to soil bacteria, fungi, and microscopic organisms that colonize the bone after the person died.
So you're mostly sequencing the dirt, not the human.
Exactly. Whole genome sequencing is largely a waste of time and money on highly degraded samples. Instead, the twist assay acts as molecular bait.
Bait!
Yeah, the researchers synthesize millions of tiny, specific sequences of human DNA that they know act as markers for different populations. They literally drop these magnetic baits into the genetic soup extracted from the bone.
And the human bait only latches onto the ancient human DNA, just completely ignoring all the soil bacteria.
They've got it. They then use a magnet to pull the bait out, leaving all the bacterial junk behind. In this specific study, they targeted over 1.3 million single nucleotide polymorphisms, or SNPs.
Let's break down SNPs for the listener. These are basically the tiny typos in the genetic code, right?
Yes, exactly. The human genome is over 3 billion letters long, and the vast majority of it is identical in all of us. But occasionally, there's a single letter variation, an A instead of a C, at a very specific location. These variations are SNPs.
Okay.
Because populations that live near each other share the same typos over generations. Tracking these SNPs allows researchers to compare this ancient DNA directly against massive databases of modern and other ancient populations to see exactly who they are related to.
It's kind of like finding a unique spelling error in a book and using it to figure out which printing press made it.
That's a great way to think about it.
So after they ran all this complex chemistry and quality control, they realized 2 of their samples actually came from the exact same individual, so they ended up with viable genetic data from 10 unique people.
10 individuals from the cave,
Yeah.
6 genetic females, and 4 genetic males.
And they determined the sex just by looking at the DNA, right?
Yes, they look at the ratio of sequences mapping to the Y chromosome versus the X chromosome. And interestingly, all the soft tissue samples that yielded DNA happened to come from the males.
Huh. Fascinating. And these 10 people weren't just a random assortment of strangers who happened to fall into a hole.
No, not at all. The genetics confirmed they were intimately connected. By comparing the number of shared allelic differences between the sequences, the researchers could actually map out kinship. They identified one individual who was a second-degree relative of another.
So second degree, that would be like an aunt and a niece, or half-siblings, or a grandparent and a grandchild.
Correct. And they found another pairing that were second to third-degree relatives, which pushes out to first cousins.
So the picture coming into focus here is a close-knit group, a family or an extended family unit buried together in a sacred communal tomb. That all tracks logically with what we know about human behavior.
It does.
But here is where this deep dive takes a massive left turn. When the researchers finally looked at the ancestry of these 10 people, mapping those SNPs against the databases, it didn't match anyone living in the area today. It didn't match anyone they had ever seen before.
What's fascinating here is the sheer isolation of their genetic signature. It's the real crux of this paper. To grasp why their DNA was so shocking to the researchers, we really need to talk about a concept known as the Indian cline.
The Indian cline. Okay, I want to make sure you and I really understand this because it seems central to the mystery.
It really is. It is the foundational map of genetics in South Asia. Imagine a vast genetic spectrum stretching across the subcontinent. On one far end of this spectrum, you have populations that carry a higher proportion of ancestry related to West Eurasian, so people from Iran, Central Asia, or even further west.
Okay, got it.
In modern demographic terms, groups like Pathans or North Indian Brahmins cluster near this end of the spectrum.
So that's the West Eurasian end of the line.
Now look at the complete opposite end of the spectrum. Here you have ancient indigenous South Asian populations with minimal to zero West Eurasian genetic influence. The researchers in this paper used the modern Paleyar people from southern India as a proxy to anchor this far end of the cline.
So we have two distinct poles.
Yes, and here is the key. The vast, overwhelming majority of people living on the Indian subcontinent today and in the past fall somewhere along the line connecting those two poles.
So they're a mix.
They are a mixture, but it is a predictable mixture. If you map their DNA, they land smoothly somewhere along this specific gradient, this Indian cline.
But when they mapped our cave people, they broke the map.
Completely. The researchers used a statistical tool called Principal Component Analysis, or PCA. Think of PCA as a giant multidimensional map. The algorithm takes those 1.3 million data points, the SNPs we talked about earlier, and mathematically collapses them into a two-dimensional graph.
Like a scatter plot.
It's exactly like a scatter plot. Physical distance on this graph represents genetic relationship. If two dots are close together, those people are closely related.
And when they plotted the individuals from Old Lady Spider Cave onto this giant map of South Asian genetics,
They did not land anywhere near the Indian cline.
they were way off in their own bizarre corner of the map.
They formed an incredibly tight, homogeneous cluster, completely separate from the typical South Asian gradient. The math showed they fell on a totally different line. A line connecting modern and ancient Tibetans on one side to a very specific point on the Indian cline represented by North Indian populations like the Dogra and North Indian Brahmins on the other.
Wow. And when the software calculated exactly what these cave people were made of, it was a massive shock. They were an almost mathematically perfect 50-50 split.
It was a startlingly precise mixture. Roughly half of their ancestry came from a population well proxied by present-day North Indians, and the other half came from a population genetically identical to ancient Tibetans.
Half North Indian,
Yeah.
half ancient Tibetan. A perfectly clean 50-50 split.
Yeah.
When I was reading this, I kept trying to think of an analogy for how strange this is. I think the best way I can describe it is like, imagine discovering an isolated, hidden mountain town, right? And you sit down at their local diner, and for centuries, their traditional everyday meal is a perfectly balanced Mexican-Japanese fusion dish.
Okay, I like this.
Right. Like an equal mix of both culinary traditions, refined over generations, but totally isolated from Mexico or Japan. You would be looking at this plate, asking yourself, how did this specific, perfectly balanced blend happen here? And why hasn't it been influenced by Italian food or French food or literally any other passing traveler?
That culinary analogy hits the nail on the head because it highlights the core mystery the researchers faced. It is not just that they are a mix of two populations, it is the cleanliness and the rigid balance of that mix that basically defies logic.
Which is exactly what threw me for a loop. Let's think about the geography of Ladakh for a second. The paper points out right at the beginning that Ladakh is essentially the ultimate geographic crossroads of the ancient world. It connects the Tibetan Plateau, Central Asia, the Tarim Basin, the Hindu-Kush-Pamir corridor, Kashmir, South Asia. I mean, it is a massive hub.
It really is. It has been a major highway for trade, migration, and conquest for millennia. The archaeological record proves this unequivocally. If you look at the rock art in the region, you have Bronze Age petroglyphs showing clear cultural links to South Siberia and the Altai Mountains.
Right.
You have Iron Age steppes-style carvings linking to the Saka cultures of the Central Asian steppes.
You have ceramics from the Hellenistic period showing Greek influence, Kushan inscriptions. It's a revolving door of humanity. So if Ladakh is this bustling intersection of a dozen different ancient cultures and trade routes, my assumption is that the DNA of the people living there should be a messy, chaotic genetic soup of dozens of groups.
Right, that would be the expectation.
Why on earth is it this clean, isolated, 50-50 split between just two specific populations?
Well, you're getting at the core of why this discovery is so scientifically precious. The cleanliness of this genetic split implies something profound about how their society functioned. If they were living in a bustling crossroads, constantly interacting and intermarrying with passing traders and nomads, their DNA would be a mosaic of a dozen different ancestries.
You'd be all over the map.
Exactly. The fact that it is a clean 50-50 split means they were a highly isolated, stabilized population.
They weren't taking in newcomers.
Precisely. They mixed once between these two specific groups a very long time ago, and then they closed their doors. They formed their own distinct society, their own closed gene pool, and they maintained it rigidly without any significant outside genetic influence for generations.
Which naturally leads us to the next massive question in this deep dive. When did that original 50-50 mix actually happen? We know that 10 people in the cave died around 1,500 years ago. Did their immediate parents meet, like one North Indian traveler, one Tibetan local, fall in love and have these kids? Was it a recent first-generation fusion?
To answer that timeline question, the researchers couldn't just look at what the DNA was made of. They had to look at the physical shape of the DNA itself. To do this, they used a highly specialized software program called DATES.
DATES. I love when scientists force an acronym to sound like what they're studying, dating the ancient relationships or something.
It's a classic move, yeah. It stands for Distribution of Ancestry Tracts of Evolutionary Signals.
Okay, quite a mouthful.
It is. And what DATES does mathematically is measure something called admixture linkage disequilibrium.
Okay, let me stop you right there. Admixture linkage disequilibrium. That is a massive block of dense jargon. If we are going to understand this time machine, we need to translate that into something we can visualize.
Fair enough. It is a complex biological mechanism, but the underlying concept can be visualized quite clearly.
Let me try pitching an idea, and you tell me if I'm on the right track. I want to use a deck of cards.
All right, let's hear the card metaphor.
Imagine you have two brand new, completely unshuffled decks of cards. One deck is entirely red cards. Let's say that deck represents the pure ancient Tibetan ancestry. The other deck is entirely black cards. That represents the pure North Indian ancestry.
Okay, I'm trekking with you.
Now, generation one. A Tibetan and a North Indian meet and have a child. That is the first shuffle. You take the red deck and the black deck and you shuffle them together exactly one time. If you lay that deck out on a table, what does the resulting deck look like?
If you only riffle shuffle it once, you're going to have big, unbroken chunks of red cards next to each other, followed by big, unbroken chunks of black cards.
Yes, large, intact blocks of ancestry. Now that child grows up and has a child of their own with someone else from this newly mixed community. That is generation two. That is the second shuffle. What happens to those big, solid blocks of red and black?
They inevitably get broken up. The big blocks of red and black get split into smaller blocks.
And with every single generation, every new human born, every new shuffle of the genetic deck, those blocks of red and black cards get broken down into smaller and smaller and smaller individual pieces.
That is actually a brilliant way to explain admixture linkage disequilibrium.
Yeah.
The biological mechanism behind your shuffle is a process called recombination. When a human body creates sperm or egg cells, the chromosomes from their mother and their father literally pair up, physically cross over each other, and swap pieces of genetic material.
So they're physically cutting and pasting the code.
Yes, essentially. So when two distinct populations first mix, the first-generation offspring inherit entire unbroken chromosomes from each ancestral group. But over time, because of this recombination process, the long, continuous segments of DNA that trace back to one specific ancestral group get chopped up into shorter and shorter fragments.
Makes sense.
And the software DATES literally measures the physical length of those ancestral DNA segments in a unit called centimorgans.
So the logic here is, if the researchers look at the DNA and see really long segments of Tibetan and North Indian code, the mixing happened recently, only a few shuffles. But if the segments are super tiny and fragmented, the mixing happened a very long time ago. Lots of shuffles.
Exactly. The software reveals a clear covariance of ancestry with distance. And when they measured the lengths of the segments in the old Ladakh individuals, the data fit a perfect single exponential decay curve.
Explain the significance of a single curve.
It means they did not have multiple ongoing waves of mixing over centuries. They were insulated. It was a single massive mixture event between two groups that then decayed steadily, without interruption, over time. And based on the incredibly small size of those segments, DATES calculated that the original mixing event, the very first shuffle of the deck, happened roughly 50 generations before these specific people were buried in the cave.
Wow, 50 generations.
Yeah. The researchers mathematically assumed an average of 29 years per human generation. If you multiply 50 generations by 29 years, you get 1,450 years. Add a little statistical margin of error for the software, and they estimate the initial admixture occurred approximately 1,343 years before the date the individuals actually lived.
Okay, let's do the math on the global timeline here, because this is the real aha moment of the paper for me. The people whose bodies were found in the cave died around 1,500 years ago, but their ancestors, the specific individuals who originally combined the North Indian and Tibetan DNA, mixed together 1,300 years before that death date.
Which puts the formation of this unique 50-50 population at roughly 2,800 years before the present day. That lands us right around 800 BCE.
That is staggering to think about. This perfectly balanced 50-50 society survived, culturally and genetically intact, completely isolated from outsiders, for over a millennium before the individuals in the spider cave were even born.
It completely rewrites our understanding of the stability of human populations in this volatile region. We often think of geographic crossroads as places of constant genetic churn and chaos. But here is hard biological proof that a group could form, find a niche, close their ranks, and exist in a state of sheer genetic stasis for over 1,000 years while entire empires rose and fell in the valleys below them.
They were just doing their own thing, living up in the clouds. And speaking of living in the clouds, I really want to talk about the physical toll of that environment. 4,000 meters is no joke. I get winded walking up a steep flight of stairs. Living your entire life for over 1,000 years at that extreme altitude requires some serious biology.
Oh, it requires extreme physiological adaptation, or you simply will not survive to reproduce. The oxygen partial pressure is so low that a normal, unadapted human body struggles to function at a cellular level.
Yeah.
You can develop severe hypoxia, acute mountain sickness, and in severe cases, fatal fluid accumulation in the lungs or brain. It is a harsh, actively hostile environment for a primate.
But we know that modern Tibetans thrive there. They live their whole lives at that altitude, and they thrive because of a very specific genetic trick hidden in their DNA, right?
Yes. Modern and ancient Tibetans frequently carry a specific genetic allele. It's a variant of a gene called EPA1 that confers profound adaptation to a high-altitude environment. Essentially, it regulates how the body produces red blood cells.
Okay, how does that work?
When normal people go to high altitude, our bodies panic and overproduce red blood cells to try and grab more oxygen. But this makes the blood incredibly thick and sludgy, which causes heart attacks and strokes.
Yikes.
Yeah. The EPAS1 variant prevents this dangerous overproduction, keeping the blood flowing smoothly, even in thin air.
And here's the craziest part of that biology. Early humans didn't evolve that gene on their own. They inherited it from a ghost species.
They acquired it from the Denisovans.
The Denisovans. For anyone who doesn't follow paleontology closely, these are extinct archaic humans, evolutionary cousins of the Neanderthals.
Exactly. Sometime in deep prehistory, as early modern humans were migrating through Asia, they encountered and interbred with Denisovans. In doing so, they picked up this specific, highly advantageous piece of DNA.
Wow.
It is a textbook example of what evolutionary biologists call adaptive introgression. The gene was so incredibly useful for surviving on the high Tibetan plateau that natural selection rapidly pushed it through the population. The math is pretty simple. If you had the Denisovan gene, you survived, had healthy pregnancies, and passed it on. If you didn't, you struggled to reproduce at altitude.
So let's look at the researchers' thought process. They are studying our old Labi spider cave mummies. These people are living at 4,000 meters. They have been living there as a staple population for 1,000 years. And crucially, they are 50% Tibetan.
The researchers must be thinking, jackpot, we are going to find this Denisovan supergene dominating their DNA.
It is the absolute most logical biological assumption you could make. If half your ancestry comes directly from a population that carries this gene, and you live in the exact extreme environment that makes this gene a matter of life and death, you expect evolutionary natural selection to have amplified it over those 50 generations. You would expect almost every single person in that cave to carry it.
But when they looked, they didn't find it everywhere.
No, and the twist here is profound. They tested the raw genetic data from these 10 individuals, specifically hunting for 11 distinct SNPs that are diagnostic for this Denisovan haplotype. Because ancient DNA is fragmented, they weren't able to get a perfect read on every person, but they were able to get reliable reads on 9 specific chromosomes from the group. And out of those 9 readable chromosomes, how many actually carried the Denisovan high-altitude adaptation?
Only one.
Just one single chromosome out of 9. So what does this all mean? If you are living in the clouds, shouldn't natural selection make that high-altitude gene absolutely mandatory for survival? Why didn't it spread through their population like wildfire over a millennium?
Well, if we connect this to the bigger picture, this finding tells us something fascinating about how human adaptation actually works in the real world. The researchers ran the statistical probabilities on this. They estimate that about half of ancient Tibetan chromosomes carry the haplotype.
Okay.
Since the old Ladakh people are exactly 50% Tibetan, you'd expect about a quarter of their chromosomes to have the gene just by random chance, assuming no natural selection was happening at all.
Right, 50% of 50% is 25%.
Exactly. And out of 9 chromosomes tested, 25% is roughly 2.25. So seeing only one chromosome with the gene isn't a massive, impossible statistical anomaly. It is entirely within the realm of random chance. But the key takeaway for the scientists is what we don't see. We don't see the gene being overwhelmingly selected for. We don't see 7 or 8 out of 9 carrying it.
It wasn't dominating their biology.
The paper concludes that after this 50-50 admixture occurred, this specific Denisovan haplotype simply was not under strong positive selection in this specific group.
But how is that practically possible? I mean, they were at 13,000 feet. You can't just ignore a lack of oxygen.
There are 2 main possibilities, and both are fascinating. One is cultural adaptation. Humans are incredibly innovative. Maybe this society had developed specific social practices, unique housing structures, or specific diets that mitigated the harshness of the environment so effectively that the biological imperative to adapt was softened.
So they found a cultural workaround to a biological problem.
Exactly.
They essentially said, we don't need a genetic supergene. We have really warm yak coats, great fires, and a system for managing exertion.
That's entirely possible. The other possibility is biological, but hidden. Remember, they are 50% North Indian. It is entirely possible that the North Indian side of their genetics provided a different, previously unknown biological mechanism for coping with the altitude, making the Denisovan gene redundant. Evolution is remarkably messy, and there is rarely only one single genetic pathway to solve a biological problem.
It just shows that we can't always predict how genetics will play out, even in extreme environments where the rules seem obvious. But there was one more bizarre clue in their DNA that I want to talk about. A total curveball that seemed to come out of nowhere, and it has to do with the mitochondria.
The mitochondrial DNA. Yes, this is perhaps the most geographically baffling detail in the entire paper.
Since mitochondrial DNA traces the direct maternal line without the messy shuffling of nuclear DNA that we talked about earlier, what did the maternal haplogroups, the branches of the maternal family tree, actually show us about these people?
Well, most of what they found made logical sense. Some individuals carried South Asian haplogroups like M65A or U2B1, which they inherited from the North Indian side. Some had Tibetan haplogroups like F2G or D4J1B. That is exactly what you would expect from a 50-50 mix.
Right.
But then they looked closely at a pair of relatives in the cave, specifically the second to third degree relative pair. And they share a maternal mitochondrial haplogroup designated as U5A1D1.
U5A1D1. Where exactly is that lineage supposed to be from?
It is deeply characteristic of modern and ancient northern Europeans. Specifically, it is found in populations originating around the Caucasus Mountains and the lower Volga area of Russia.
Russia, Northern Europe. What on earth is a maternal lineage from the Russian steppes doing inside a mummified body in a cave 4,000 meters up in the Himalayas?
It is a stunning geographic leap, but it reveals the sheer scale of ancient massive human migrations. The researchers hypothesized that this specific genetic marker hitched a ride via the ancient steppe migrations. Thousands of years ago, nomadic pastoralists from the Eurasian steppes, often referred to as the Yamnaya or related cultures, began a massive expansion, migrating south into Central Asia and eventually pushing all the way into the Indian subcontinent.
This is the migration that brought Indo-European languages and a huge wave of new genetics into South Asia, right?
That is the one. This U5A1D1 haplogroup likely arrived in North India as a very low-frequency genetic marker carried by a single woman or a small group of women with steppe ancestry. She passed it down her direct maternal line, mother to daughter, generation after generation in North India, until eventually one of her distant descendants became part of that original 50-50 mixture event 2,800 years ago.
And from there, it was passed down for another 1,300 years, completely isolated within this mountain group, until it ended up in Old Lady Spider Cave. I mean, the sheer expanse of time and distance is dizzying. A tiny genetic echo of a woman from the Russian steppes, frozen in the Himalayas a millennium and a half later.
It truly highlights how interconnected human history is, even when populations seem incredibly isolated at a first glance. The DNA remembers the entirety of the journey, even if the people themselves have long forgotten their origins.
Okay, so that brings us to the ultimate fate of these people. What happened to this perfectly balanced 50-50 ghost population? Did they just vanish into thin air? Did their society collapse? And this cave represents the last few survivors freezing to death?
No, the genetic evidence proves they didn't disappear entirely. They did not hit a genetic dead end. To find out what happened to them after the period of the cave burials, the researchers did something very clever with their statistical modeling. They took their best-fitting mathematical model for the old Ladakh population, the strict 50-50 mix, and they started adding modern Himalayan populations into the software to see how the model reacted.
Oh, so they were checking to see if anyone alive today is a match for the ghosts.
Exactly. And they found that their isolated model failed in a very specific, revealing way when they added groups like the Monero and Balti people who live in the Ladakh region today.
Why did the model fail? Like, what does a failure mean in this context?
It failed because the software detected shared genetic drift between those modern groups and the old Ladakh individuals. In statistical terms, a failure of the isolation model here is actually a massive success for tracking their legacy. It means the old Ladakh people are not completely separate from the modern populations. The ancestors of the modern Monero and Balti people received significant gene flow from the old Ladakh population.
Oh, I see. So the ghost population didn't die out. They just, well, dissolved into the broader population.
Yes. Eventually, after a millennium of strict isolation, their cultural or geographic borders became porous. For whatever reason, they started mixing with other groups arriving in the region. The paper specifically notes that modern Monero people can be modeled as having about 56% of their ancestry from an old Ladakh-related group, and the Balti people have about 45%.
So their legacy is very much alive. The descendants of the people of Old Lady Spider Cave are still walking the Himalayas today, just woven into a larger, more complex genetic tapestry.
It is a profound realization of the scale and resilience of human history. You have a unique group that forms from two distinct worlds, North India and Tibet. They isolate themselves completely for 1,300 years. They bury their dead, carefully mummified in a freezing, inaccessible cave. And then, slowly, over the next 1,500 years, they blend back into the broader human family.
It makes you wonder how many other ghost populations are out there, completely unknown to history, just waiting for the right cave to be discovered.
Undoubtedly many. The human genetic landscape is incredibly complex, and we are only just beginning to scratch the surface, especially in regions like South Asia, where the climate usually erases the evidence before we can find it.
But there is one final lingering mystery from this paper that I cannot stop thinking about, and it's a detail the researchers explicitly point out at the very end of their discussion. We keep calling them the old Ladakh people. We found them in Ladakh, but the researchers admit we don't actually know if they formed in Ladakh.
Right. That is the ultimate frustrating caveat of ancient DNA. Finding a body in a specific place only tells you exactly where that person died. It does not tell you where their ancestors lived 1,000 years prior.
Because we know the admixture, that very first shuffling of the genetic deck, happened 2,800 years ago. The researchers suggest that this perfectly blended society might not have formed in Ladakh at all. What if the mixing happened somewhere else entirely, like modern-day Nepal?
It is a highly plausible scientific scenario. The geographic borders and population distributions of the ancient world were absolutely nothing like the borders we know today.
Just imagine that for a second. What if this ancient, perfectly balanced 50-50 society formed in an isolated valley in Nepal? And then for some reason, maybe a changing climate, a violent conflict, or just a search for better high-altitude pastures, they packed up and started moving. A nomadic tribe, completely insular, wandering the treacherous high-altitude peaks of the Himalayas for 1,000 years. Moving from valley to valley, generation after generation, never mixing with outsiders, carrying their unique genetics, their Denisovan ghost DNA, their steppe echoes, all the way across the roof of the world.
Until finally, after 13 centuries of wandering, they arrived in the region of Ladakh, established themselves, and eventually laid their dead to rest in the freezing dark of Old Lady Spider Cave.
It completely reframes the entire story for me. It wasn't just isolation in a single valley. It might have been an epic, millennium-long migration across the most hostile terrain on Earth. And it leaves you with this incredible, unanswered question. Where did their journey truly begin?
And perhaps more intriguingly, what forced them to finally stop?
It's a mystery that the DNA can't quite answer yet. But maybe, just maybe, there's another cavey out there, waiting to be found.
Heliox is produced by Michelle Brucker and Scott Bleakley. It features reviews of emerging research and ideas from leading thinkers, curated under their creative direction with AI assistance for voice, imagery, and composition. Systemic voices and illustrative images of people are representative tools, not depictions of specific individuals. Thanks for listening today. 4 recurring narratives underlie every episode: boundary dissolution, adaptive complexity, embodied knowledge, and quantum-like uncertainty. These aren't just philosophical musings, but frameworks for understanding our modern world. We hope you continue exploring our other episodes, responding to the content, and checking out our related articles at helioxpodcast.substack.com.